Well, hello, and let's try something new. We're going to call this the immune system guinea pig, and we're going to see how this goes. Your text, in our text, we spoke immediately about what's the basic instruction of the actual lymphatic system itself, right? So let's take a look here at the lymphatic system according to this open educational resource document, meaning this is freely available for us to use. And from my perspective, I went through a large number of things to try and find something that we could use sort of regularly that will be helpful to you. And they leave out the thymus, so let's draw in a thymus for you here. I'll even use their green. Let's see if I can use their green. Find a green for you that looks like theirs. Look, there's a green, and I'm going to put a thymus in front of this right here because it's going to be important when we think about where T cells develop later inside chapter 22 inside our textbook, right? But this general idea that it's going to collect materials from the interstitial fluid inside your body to then route backwards through an open circulatory system. Let's make sure everybody gets that. Over here, lymphatic vessels are an open circulatory system, which means the central idea here is pressure is going to push fluid into these, and they'll then draw back into the lowest point of pressure, which would be your cisterna chyli here, for the lower parts of the body, then moved up and then put back into your return circulatory system, which is why this figure has these major blue vessels, the thoracic duct, the right lymphatic duct here feeding into the actual blood vessels that are then going to the superior or inferior vena cava. Well, superior vena cava. I stand corrected. Now, there are four major lymph nodes that if we were in lab, I'd say, hey, look, I'm going to point an arrow to here, and I'd expect you to be able to tell me that that's the patient's left inguinal lymph node cluster, or this is the patient's right cervical lymph node cluster. And you know this because when you were a kid, or you yourself now are sick, somebody will palpate those areas, and if you're sick, they tend to become inflamed. Okay? Let's get rid of that. Let's move on to the next one here. And the major filtering structure inside of your body is this thing that's called a lymph node. And historically, I have people learn how to draw these. So here's a very simple cartoon of a lymph node. It has a capsule in green for you on the outside here. And then it has this hilus. And what this hilus represents is an area, get this, of low pressure. So materials are going to flow in here. You'll see how in a moment. And then they're going to filter over populations of cells that are occupying the cortex, traveling into the medulla, okay, and then exiting out through this hilus. So these would then be, you'll see in a moment, afferent lymphatic structures, and therefore these are efferent lymphatic structures, which only makes sense when you think about what we've learned in the past. So you have a pressure regulating system here. That's the purpose of the capsule. And to have this hilus is to represent the area of lowest pressure. Get rid of the artwork. And let's move on to the... In here you can see now what they've done is they've put in the structures that are inside the cortex. Remember, in the previous video I showed you guys, I said in class, I'm definitely going to expect that you know that you have antigen-presenting cells here, which will be the dendritic cells or macrophages. And then you'll have B cells primarily, right? And those B cells are going to be looking for materials that will activate them. And then we'll have some population of T cells. And all of a sudden you can see we've moved here from the cortex to the medulla, just like I said we would. Okay. And these germinal centers you're going to see, they actually have vasculature. And that's something that your textbook misses to discuss. And you can see that traveling between each one of these, you'd actually have germinal centers, which means not only do you have materials that are flowing inward here, we have vascular mechanisms to actually support these cells for living. But also think about this. Remember inside chapter 21, although we haven't really discussed it really all that well, you can get materials in and out of the circulatory structures here. Now, how does this all maintain its shape? You have these things that are called trabeculae, and these are dense irregular connective tissue that are moving inward, creating this shape. Okay, and then you can actually use some reticular tissue, which is going to solidify it, so you end up making these cortical and medullary sinuses. And remember, a sinus is an area of lower pressure, so therefore everything still makes sense. Inside your body as well, if we're looking now, say, inside an intestinal structure, you can see here the appendix is off over here. We'll talk about that later. But the associated lymphatic tissues, here we have this Peyer's patch, and in one of the figures I'll show you, I'll put together for you as a PowerPoint slide sort of quiz thing, will be lymphatic vessels are actually screening the materials that you're absorbing as you're digesting materials. And you'll see this is a general idea we find throughout the body. We look inside the mouth and oral structures. You have these things that are called Waldeyear's rings, which make up your tonsils. And it's called a ring because you have a lingual, 2 palatine, 2 tubal, and 1 pharyngeal. And that pharyngeal is also what you call the adenoids. And that's what actually gets swollen when you have a sore throat. All of these can become swollen, don't get me wrong. Okay? But what they're doing, of course, is they are literally screening for pathogens as materials could either be entered into your respiratory system or entered into your digestive system. So, what does a tonsil actually look like? Crypts are these areas that allow materials to move in there by migration, okay? And a germinal center is where you're going to actually have materials that are being presented to cells that occupy them. And this is distinct if we look over here. If this is the capsule of a lymph node here, and that's a germinal center, you're going to see that there's a repeating theme in all of these histological presentations, where we have some form of a germinal center of cells that are made up primarily of B cells, surrounded by some mixture of antigen-presenting cells moving in here to present materials to those cells, and then some mixture of T cells that will then move off and do some other responsibility. And you'll see what that is later. So if here's our lymph node, we can see here inside of the colon, these things that are called Peyer's patches, we just mentioned to you a moment ago. Notice they also have this sort of repeating germinal center familiarity to them. Okay? The idea, of course, is once you learn how a lymph node functions, you generally understand how all of the immune presentation mechanisms will work. This last idea here, okay? If this is the appendix, okay, and you have lymphoid tissue here, and that's mucosal, it means not only does it have the responsibility of maintaining the correct amount of bacterial flora and fauna, it also has the ability to determine how materials of the immune system will move into and out of the immune circulatory system. So once a T cell's been activated, or once a B cell's been activated, how does it make sure it becomes activated properly? And where does that fluid travel through? If we're looking here, say, for that lymph node, remember the efferent vessel that exited earlier. You can see here the same idea is happening here. Now, inside this model mock-up, you can see in red, illuminated here, is that spleen. And one thing I want you to pay attention to is during this sort of like weird time where you guys are at home and I'm here and I'm trying to figure out how to test you on all of these things, one thing I'm going to be doing is thinking to myself, how can I encapsulate multiple pieces of information into, say, every single question? So if I'm pointing at, say, during a practical, a model, an image of a model, I might be saying, okay, what is this structure? What system do I find this in? So very reminiscent of AMP1. But then it might be, what vasculature supports the spleen? So in your list of arteries and veins you were supposed to learn, you remember there's a splenic artery and a splenic vein. And then on your list of structures, you remember there's a gastric impression, so this would be the gastric impression here. But right now what I want to do is I want you to see that if you can see the white pulp and you see a trabecular structure and you can see some red pulp surrounding it, these are the histological markers that we want to make sure you see that will tell you this is a spleen. Okay? Now, I tried to use the images from your textbook. I've given you a different piece of histology here. Maybe working these in conjunction, we can put the whole story together. Okay, so that's the end of this OER, Open Educational Resources. These materials I went looking for, over the last week, trying to find the best quality materials for us to use as a lab book in conjunction with the lab sheet you have, or sheets that go along with the course itself. Now, I'm not going to have the ability to collect your lab sheets for this lab practical, number one, but number two, I'm also not going to have the ability to get you another set of these things. So I have to come up with something that's going to make sure you do this, and I'm working on that, and we'll start rolling that out over the weekend. So this is our guinea pig. I'm trying to figure out if it worked well, it didn't work well. Shoot me an email message back and let me know, okay? Take care. Have a nice day, and wash your darned hands.